On to our second presentation. This comes from Lillian Wallis, who's currently a master's student at the University of Bath, but she's been doing a placement in Kim Dora's lab in the Department of Pharmacology at the University of Oxford. And today she's going to tell us about the involvement of TRP channels in endothelial-myocardial interaction. So, Lillian.
Yes, hello. Thank you, Nicola.
So if you'd like to start, share your screen.
Okay, so thank you everyone for listening to me today. So my name is Lily Wallis, and like Nicola said, I am a third year undergraduate from the University of Bath, but doing my placement with Kim Dora at the University of Oxford. So the data that I've collected today is just some preliminary data, but I'll talk you through it when we get there. So we have been looking at the relationship between the endocardial endothelium and the myocardium within the heart. And we're specifically looking at the role of TRP channels in this signalling. And we used a novel technique which allowed us to do this. So the intracardiac endothelium is the innermost layer of the heart, and its structure is thought to be analogous with vascular endothelium. And due to the well-established feedback relationship between vascular endothelial cells and smooth muscle, it follows that there would be the same kind of feedback mechanism within the heart, so involving things like nitric oxide and prostacyclin and endothelin-1. And we know that vascular endothelial dysfunction often precedes cardiac disease. So we also think that dysfunction of the endocardial endothelium might precede the development of the same kind of cardiac diseases. So the novel method that we have been using allows us to look at calcium handling in intact endocardial endothelial cells. And we're also able to see the calcium handling in myocytes, which has never been done before. So we have a lot of scope for looking at how the effects of drugs might alter calcium activity, and we can also measure intracellular, subcellular calcium events, and we've got the ability to measure calcium activity as well as changes of tension. So just an overview of the method. So the first thing we do is dissect a pectinate muscle column from the right atrial appendage of a rat. So this is carefully dissected out and then mounted in a confocal imaging chamber with the endocardial surface downwards. And then we load the strip with calcium indicator. And if you look at the bottom image here, you can see we've got the endocardial endothelial cells. And then just below them, we've got the atrial myocytes. If you imagine the column's been tilted slightly. And then in the top image, we have got some immunolabeling, which has characterized the cell types here. So we've got the blue endocardial endothelial cells, yellow nuclei, and again, we've got the red myocyte. So this is a proposed mechanism of action for how these two cell types influence each other. And we're interested in TRP channels specifically. So they are calcium selective and can upregulate the production of endothelin-1 and also upregulate the production of nitric oxide, which both have opposite effects on the cardiomyocytes. And there was a 2017 paper which confirmed the presence of TRPC6 channels in the endocardial endothelial cells. So what we're hoping to do is characterize more TRP channels and then in the future look at some of the mediators that could be released from the endocardium. So here we've got an experiment where we added 1 micromolar of ACH. So I'm just going to show you a video of the calcium increase following that addition. So here you can see the endocardial endothelial cells. And that was a significant change when we compared it to the baseline calcium before any drug was added. And then in this video, we've got the effect of ACH on our myocytes, and we didn't see it have an effect. There was no change in the frequency or the amplitude compared to baseline. And then a similar thing was found when we added ATP, which activates purinergic receptors. So again, we had this increase in intracellular calcium of our endocardial endothelial cells. And again, there was no change in the frequency or the amplitude of the myocyte calcium transients. And here we added a TRP-C3 and a TRP-C6 agonist, GSK170. So it's thought that low concentrations of this agonist activate TRP-C3 channels, so 1 micromolar. And then if we increase this to 3 micromolar, we are activating TRP-C6 channels. So here we added both concentrations. So the lower concentration didn't seem to have any effect on our endocardial endothelial cells, but our high concentrations did seem to cause an increase in calcium. So we think that there might be TRP-C6 channels present here. And when we look at the myocyte calcium activity, when we added 1 micromolar of this GSK, we did see an increase in the amplitude of our calcium transients. So here's some immunolabeling of our 2 different cell types. So we can see that TRP-C3 expression is mainly within the myocardium, so the pink, and then TRP-C6 is expressed in both the endocardial endothelial cells, so this yellow, and then we've got punctate expression in our myocardium. And to confirm our cells were viable, we applied electrical field stimulation. So before we stimulated our cells, so this is the myocytes this time, we had unsynchronized calcium activity, but as soon as we applied the EFS, we had a lovely increase in our global calcium. And then a slight period where our cells were recovering, and then we had that same unsynchronized calcium activity start up again. So this suggests that our cells are viable and that they're healthy and behaving as they should. And this EFS could also be used to determine the effects of agonists on electrical signaling, so things like recovery time after EFS is applied. So overall, we have developed a novel technique that allows us to investigate endocardial endothelial cells as well as cardiac myocytes. And we have confirmed the presence of TRP-C6 channels. And we could apply this technique to different regions of the heart. So the SA node, for example, to have a look at calcium signaling in those areas specifically. And because we've optimized and developed this technique, we're now able to repeat our experiments and get more ends. So the results I've shown you are based on a very limited number of ends, but we hope to go forward and get some more data. We'd also want to investigate the role of endothelin-1 in the endocardial endothelial myocyte signaling mechanism. So we would use immunohistochemistry to localize endothelin-1 immunoreactivity before and after we add our TRP agonist to see how the TRP channels might affect endothelin-1 release. And we could also add endothelin-1 to measure changes to our endocardial endothelial cell and also our myocyte calcium handling. And we'd also want to compare data from healthy animals to data from animals with diseases such as hypertension and cardiac hypertrophy, where we've seen that there is an upregulation of TRP channels. So we could investigate the effects of that upregulation a little bit more. Okay, that's me done. So I would like to thank Kindle and Chris Garland and everyone else in the group, with special thanks to Luda Borisova, who has helped me to learn all of the methods. Thank you.
Thank you very much, Lily. That was a lovely presentation. You got the videos to work, which was excellent, and some really, really interesting data and a novel way to look at these TRP channels. So, does anyone have any questions for Lily via the chat or by raising your hand if we've solved that issue? No, not for the moment. So maybe I could ask you, Lily, from your reading and other published studies, what's the evidence that there's a differential distribution of TRP channels across different areas of the heart, either in mice or rats or humans?
Okay, so I've read that TRPC6 channels are upregulated in like cardiac hypertrophy specifically, and also in hypertension. So the immunohistochemistry that I showed you was actually from hypertensive rats. So I would have to repeat that to have a look at expression in like a normal phenotype, and again in hypertension and potentially other disease states. Yeah,
from different areas, I guess, of the heart. Yeah. Okay, so we have some questions now. So one from Casper Hempel. How long can you keep the cells alive in the chamber, and how do you assess this?
Oh, okay. So from dissection to the time we finish the experiment, it can be a few hours, so I'd say up to 3 hours. And by doing the EFS, that's how we can determine if the cells are still viable and if they're still behaving how we would expect them to.
So presumably a proportion, or a fairly significant proportion of that time, is doing the dissection and getting the cells prepared into the chamber.
Yeah, I mean, it doesn't take too long, so it probably only takes about 10 minutes to dissect and then mount the strip. So we are able to do the experiments fairly quickly. So I don't think there'd be any issue with them dying in that time.
Good, thank you. Another question, how do you identify the SA node cells using this method?
Well, I'm not too sure. I don't think that we have looked at SA node cells yet because we haven't got any action potentials forming. So we're able to look at contractility of the strip as well as the calcium signaling in individual cells. And so far, we haven't actually seen any changes in tension when we add our agonist, or when we add our ACH or our ATP. So that might suggest that we haven't got any pacemaker cells in the strip.
Okay, that question was from Nina. So if you have any further thoughts on that, perhaps pop it into the chat. And the final question is from Charlotte Buckley. I was just wondering whether you get beating of the myocytes.
So no, we don't really. I've seen the appendage beating after it's been dissected from the heart, but I've never seen any contraction of the strip once it's been mounted. And like I said, we are able to measure that with a tension transducer, and we haven't seen any changes.
Okay, there is one more question in the chat, but perhaps you could answer that online in the chat facility, as we must move on to the next presentation. So thank you very much, Lillian. Good luck with the rest of your studies.
Thank you very much.